This video looks at the method of PCR, that is polymerase chain reaction. This is a method of making many copies of a DNA fragment. If we look at this diagram here, we see step 1, we have the DNA fragment which we want to make copies of. This DNA fragment is in solution with free-floating DNA nucleotides and a DNA primer. It's heated to 95 degrees in the thermocycler. This allows the DNA strand to separate into 2 single strands of DNA. We then move to step 2. Notice the temperature has dropped to 55 degrees centigrade with the thermocycler, and the DNA primers, these being complementary DNA bases, attached to the ends of the single strands. This is for several reasons. One, it allows the DNA polymerase a starting point, so it will allow free-floating complementary nucleotides to attach in stage 3, but also it prevents the 2 single strands of DNA from simply rejoining again. If we move to step 3, we can see the temperature has changed again to 72 degrees, again controlled by the thermocycler. The primers are already attached to the ends of the single strands of the DNA, and DNA polymerase is now attaching the individual free-floating DNA nucleotides. This allows us to make 2 complete strands of DNA. And then the cycle begins again. Each of these 2 strands is separated, primers are attached, nucleotides are attached, and so on, which would go from 2 to 4 to 8 to 16 individual fragments of DNA. So if we look at the mark points for this, as I say, it's a way of making many copies. We heat it at 95 degrees. We anneal the primers. Annealing simply means joining the primers. These are complementary bases. The starting point for DNA polymerase, and they prevent the strands from rejoining. Again, as I said, at 55 degrees. Stage 3, the DNA synthesis. The DNA polymerase joins the free-floating complementary nucleotides onto the DNA strand at 72 degrees, thereby completing the strand. Why do we do PCR? 2 reasons we can do PCR. It allows us to make a DNA fragment which we can then insert into a vector, into a bacteria, into a plasmid, which will allow that bacteria to then generate the product that we want from that piece of DNA. 2, we would use it for electrophoresis. By making many copies of the DNA fragment, it allows us to amplify the DNA so that when we run the electrophoresis gel, we get a good clear showing of where the fragment is on the DNA, of the DNA on the gel.